Ingredients of PCR. The major ingredients of PCR, they are microfuge tube, thermal cycler, DNA template, primers that can be left primer and a right primer, buffer that can maintain the pH of the PCR mix, magnesium chloride which is required for the activity of the DNA polymerase, distilled water, DNTPs which are also called as deoxynucleotide triphosphates, and DNA polymerase. So these are the main ingredients of a PCR mixture. In case of PCR mixture, small tubes which are called as microfuge tubes, they are used to prepare the PCR mix, and thermal cycler is used, which is also called as PCR machine, to amplify the DNA. So if we see the structure of a microfuge tube, these are small cylindrical plastic tubes with conical bottom. So this is a typical microfuge tube in which PCR reaction is performed. These tubes, they can be in the form of single tubes, but they can also available in the form of strips. Here we can see the strips where the PCR tubes, they are joined with each other. So PCR tube can be in the form of single tube, it can be in the form of a strip, and it can be in the form of PCR plate. So in case of PCR plate, there are 96 tubes joined with each other in the form of a plate. So microfuge tube is actually those tubes in which PCR reaction is performed. The next is thermocycler. Thermocycler is also called as PCR machine. Thermocycler works on the principle of Peltier effect. Peltier effect is actually by which the temperature raises and lowers down. So once if we have to increase the temperature, and during the next step we have to decrease the temperature, such a process is called as Peltier effect. So Peltier effect raises and decreases the temperature in a block. This is the block. Here we can see that this is block of the PCR. So in PCR machine or in thermocycler machine, there is a program that has been entered into this PCR machine, and once the program is entered, then we kept the tubes into the block, and this thermocycler increases and decreases the temperature as we have entered into this machine. So this is a typical thermocycler that we have seen here. There are different commercial companies that manufacture such kind of machines which are called as thermocycler. So in case of PCR, ingredients of the PCR, there is the main component which is called as DNA polymerase. So there are different types of the DNA polymerase. The most common DNA polymerase which are used during the PCR, they are tag polymerase, PFU polymerase, and Vent polymerase. Although there are other types of the polymerases which are used for polymerase chain reaction, but these are some of the common polymerases which are used to perform the PCR reaction. So here we can see how within a tube the ingredients are mixed together. Here we can see that this is a typical microfuge tube in which there are primers, nucleotides. Nucleotides are of 4 types: adenine, guanine, adenine, guanine, cytosine and thymine. So collectively they are called as DNTPs. So this is the DNA which is also called as template DNA. This is the DNA whose copies have to be prepared. So tag DNA polymerase is the enzyme that actually amplifies the DNA. All these ingredients, they are added in this conical flask, and ultimately this conical flask or conical tube is kept into the block of the PCR where the reaction starts. So template DNA, if we talk about the template DNA can be the DNA or it can be the RNA. If we have to multiply or if we have to amplify the DNA, then that DNA which is used to amplify is called as template DNA. But in case of RNA, RNA can also be the template, because if we have to perform or convert RNA into DNA, then RNA will be the template RNA. So we can perform PCR reaction on both by taking the DNA as a template, and we can also perform by taking RNA as a template, because RNA first is converted into cDNA and then that cDNA is amplified by polymerase chain reaction.